Okay, so we're just going to pick up where we left off. And I think where we left off was with eosinophils. Does that sound right to everyone? Anyway, I think it is. I guess either way, I'm going to pick up with interferons. So, okay. All right. So again, just by way of reminder, we're still in the innate immune system, talking about responses, cells, and then immunological responses that are not antigen specific in nature. So one of the proteins that our immune cells use that are part of the innate immune system are what we call interferons. So interferons are a very important class of cytokines, which we talked about. Cytokines are proteins, soluble proteins that our immune cells use to communicate with one another and also to communicate with other cells of the body. And so interferons specifically are released by all of the white blood cells that we have and also by fibroblasts. Fibroblasts are the cells that make protein fibers like collagen and the reticular fibers. Characteristics of interferons. So interferons play a really important role in helping to protect our cells against viral infections. So one of the things that interferons do is when they are released, so when a cell, a body cell, like a fibroblast, which are a very common type of cells, when those cells become infected, they will release, and I should say both infected cells and white blood cells will do this when they detect that a virally infected cell is, or that cells have been infected with a virus. So the infected cell will release interferons, and right now we're not going to worry about which specific interferons these are, but just suffice it to say that these cells will release interferons, and those interferons will bind to neighboring cells that have receptors for the interferons. And what this does is it triggers a mechanism by which the cells that bound the interferon, they kind of up their antiviral protective mechanisms. So for example, they start to make enzymes that are that interfere with the replication of the virus. And so again, they kind of you can think of these interferons as beefing up when they bind to neighboring cells that are not infected, just normal cells. They beef up that cell's protective mechanism and help to prevent that cell from becoming infected. Interferons also bind to and stimulate macrophages and natural killer cells. So they stimulate the macrophages and activate them so that they are essentially on high alert looking for virally infected cells. And then they also play an important role in activating natural killer cells. And those natural killer cells work by causing cells that they interact with that are virally infected to undergo apoptosis, which is that type of cell death that is very clean and does not induce an immune response. Okay, so as far as the specific interferons I want you to know, I want you to know basically these three different types of interferons. So the first one is gamma, this is alpha, and this is beta. So T cells and B cells make interferon gamma, which is a very important interferon for activating additional T cells and B cells. And then essentially all other leukocytes make interferon alpha, right? And then fibroblasts, they produce interferon beta. And then alpha and beta together, they play a really important role in controlling the inflammatory response. And this is a little bit counterintuitive, but interferon alpha and beta are actually anti-inflammatory in nature. So they play an important role in helping to turn off the inflammatory response when it is no longer needed. Okay, so now we're gonna move on and talk about another group of antimicrobial proteins that are a part of the innate immune response. And these proteins make up what is called the complement system. So characteristics, sorry, hold on one moment. Bring it here. So the complement system is made up of over 30 plasma proteins, and they all start with the letter C followed by a number. And sorry, everybody, hold on one moment. Jeez. Okay, sorry, everybody. It's just, okay, where were we? Okay, so complement. So again, all of these proteins start with the letter C followed by a number. And complement itself.
Sorry.
Yes?
I don't think some of us can see the PowerPoint that you're presenting. All I see is like your face or the picture.
Hold on one moment.
Yeah, if there's supposed to be a PowerPoint, we definitely don't see it.
Did you see it at all?
No, it's just like the picture of your face, like as if you weren't sharing your screen.
I saw the shared screen PowerPoint.
Yeah, we were seeing it for a little bit.
Yeah.
But now we're not seeing it.
There
Okay,
you
how's that?
Perfect.
Yeah, that's good.
Okay, so there are two pathways. There are two pathways by which complement can be activated. And the difference between them, and I guess I should say complement is somewhat similar to the coagulation cascade in the sense that it involves enzymes that when activated, then activate downstream proteins. So there are two ways in which complement is activated. The first is called the classical pathway, and the second one is called the alternative pathway. And the difference between them, the main difference between them, is that the classical pathway involves antibodies, and the alternative pathway does not. And again, all of these proteins are named. They all start with the letter C and then followed by a number, right? I don't require you to know the specific proteins that are involved in the classical versus the alternative pathway and or the structures when we talk about the membrane attack complex here in just a moment. That's made up of several of the complement proteins that come together. I don't expect you to know that, but I do want you to know that the classical pathway is activated by having antibodies that coat the surface, the antigenic surface of a pathogen, and the alternative pathway works by having complement bind directly to the antigens that are on the bacteria or the fungal cells or any other pathogen that we're talking about. So here are the ways in which complement works, or here's how complement works. So the first way, and one, an acronym you can use to help you remember this is OP-ICE, right? OP stands for opsonization, and then the I stands for inflammation, cytolysis, and elimination of immune complexes. So opsonization, think of opsonization, that word means to make something tasty, right? Opsonization in the context of complement, what happens with the complement proteins is the complement proteins will bind, can and will bind directly to the antigen on the surface of a pathogen. And then having that complement bound to the pathogen makes it much easier for the immune cells like macrophages, makes these entities much more visible to the immune system. So it essentially makes the pathogen tastier to the macrophages. Complement also plays a really important role in increasing or activating the inflammatory response. So it binds directly to cells such as mast cells, which again are basically basophils in the context of the tissues. It will bind to neutrophils and to macrophages, and it will activate these cells. And these cells are important cells that are involved in the inflammatory response. A third way in which complement protects you is by causing cytolysis. So, cytolysis. So the word cyto means cell, lysis means to break apart. So with cytolysis, what happens is that these complement proteins come together to form what is called the membrane attack complex. And the MAC, which is the acronym for that, is essentially a large pore or channel that is inserted into the membrane of the target cell, and it causes that cell to lyse because water will rush into the cell when the MAC is created in that cell, and that target cell will essentially explode. And then the last way in which complement works is through the elimination of by creating these immune complexes and helping to promote their elimination. So what happens is that you have an antibody, and antibodies can bind free or soluble antigen. We'll talk about when we get to the adaptive immune response, how T cells are not able to recognize free antigen. Antigen has to be presented to them in the context of what's called an MHC molecule. But antibodies made by activated B cells called plasma cells are able to recognize and to bind free antigen, which is what you're seeing right here. And when complement is around, complement will bind to what we call the FC portion of the antibody. And then that complement is able to dock onto a red blood cell. And so those red blood cells will shuttle this whole immune complex to lymphatic structures like the spleen or to the liver. And then once there, the immune cells like macrophages will take care of those immune complexes. Okay, so now we're going to move in to the topic of inflammation. So inflammation is one of those processes that is really important for activating the immune response, but too much of a good thing is very detrimental to us as organisms. So inflammation, acute inflammation, can and is very important for activating our immune responses, but chronic inflammation is something that is kind of a hallmark of diseases like autoimmune disease. And so that's an important overriding theme to keep in mind as we go through the process of inflammation. So here are the basic steps that happen during inflammation. So the first is the release of specific chemicals, which we will talk about. Those chemicals include histamine, which we've already discussed, which is what basophils and mast cells release when they degranulate. Other chemicals such as what are a group called leukotrienes, prostaglandins, and other factors all make up these chemicals that are released by cells when they are injured. And also cells that become infected release these chemicals. In response to these chemicals, there are vascular changes that take place. Specifically, we get what's called increased vascular permeability. And what that means is that the cells, the endothelial cells that make up the vasculature, they become leaky. The tight junctions kind of widen. And that's really important because it allows immune cells to easily leave the vasculature and to migrate into the tissues where the infection, where the chemical trail is leading them. Another important thing, if you will, process that occurs in response to the release of those chemicals is the expression of what we call cell adhesion molecules in the vasculature. So those are all, the acronym for that are CAMs. So cell adhesion molecules act as a type of cellular Velcro. And that's important because what that Velcro does is as the white blood cells are moving through circulation, it allows them to stick onto the capillaries, and that process helps to promote what's called diapedesis, which is the migration of these cells into the tissues. And then the third event that occurs during inflammation is the recruitment of white blood cells. And this is, well, and I'll show you a picture here in a moment that goes over this, but the processes that are a part of recruitment include margination. So margination is the word that we use to describe white blood cells sticking to that cellular Velcro, those cams. Diapedesis, that word I just used, which is the process of the white blood cells squeezing out of the capillaries into the tissue. And then chemotaxis, which is those white blood cells following the chemical trail to the site of injury or the site of infection. And then the fourth process involves the delivery of antibodies and what we call kinins, which are a group of proteins that are involved in inflammation and help to further increase vascular permeability. Various clotting proteins and complement, all of those also migrate as the fourth event in inflammation, migrate to the site of infection or injury. Okay, so here's a picture that goes along with what I was just trying to describe verbally that summarizes those events that are a part of inflammation. Again, you get release of chemicals from cells that are injured or white blood cells in the area. Those chemicals cause vascular changes. And one of those very important vascular changes is the expression of those cell adhesion molecules on the inside of the capillaries. Those allow for the cells, the white blood cells in circulation, to stick to the vasculature so that they can, and that's what margination is, so that they can undergo diapedesis, which is what you're seeing here, the squeezing of those cells out of the vasculature, and then they will follow the chemical trail to the site of injury or infection. And kind of at that same time, you get the delivery of various plasma proteins like antibodies and complement and kinins and clotting factors to that site to also help aid in the removal of the pathogenic substance that has gotten into the tissue as a result of the injury. Okay, so this slide up here goes over some of, or just kind of summarizes some of those changes or effects of inflammation that we've already discussed. So vasodilation and increase in vascular permeability are really important changes that take place to the vasculature that help to promote white blood cells getting to the area of injury or infection to help kind of clean up the mess and activate the immune response. So, and along those lines, the production of what we call exudate. So exudate is essentially interstitial fluid. So what we see in response to inflammation is the formation of exudate, right? And it's this fluid that is, right, again, this is why swelling is one of the hallmarks of inflammation. So because of this increase in vascular permeability and vasodilation, we get fluid accumulation in the area where the infection has taken place. That extra fluid helps to kind of wash the antigen or the pathogen and push it into wash the area and push that fluid into the lymphatic system, which helps to activate the immune response and especially helps to activate an antigen specific type of response. And then again, so acute inflammation is very important in helping us clear infections. However, if it goes on for longer than about 3 to 4 days, it starts to become problematic. And that's because we start to get the formation of, amongst other things, scar tissue. And so, yeah, kind of more on that to come in here in just a moment. But again, why inflammation is important is because it helps to activate the immune response, specifically the adaptive immune response, helps to prevent that infection from spreading elsewhere. And then ultimately helps to promote tissue repair. Again, it helps, inflammation results in scar tissue formation at the end of the day. So here are the cardinal signs of inflammation. And most people are pretty familiar with this because we've all had inflammation at some point in our life, right? So redness is due to the vasodilation that occurs as a result of inflammation. Heat also, is due to that increase in vasodilation and vascular permeability. The swelling is due to the formation of that exudate, that excess fluid that is formed. Pain receptors are stimulated during the inflammatory process. So kinins, bradykinin, for example, is getting a lot of press right now because we know that there's strong evidence to suggest that we get an out of control bradykinin response and when during the COVID-19 infection. So bradykinin is one of those proteins that helps to stimulate pain receptors. And pain, although pain that's chronic is problematic, acute pain is adaptive, right? Pain is telling us to not use, it's alerting the body that something is wrong. So it's very adaptive. And then loss of function is the fifth cardinal sign of inflammation that's oftentimes due to the swelling that's associated with inflammation. And as I keep saying, acute inflammation is helpful, but chronic inflammation is a process that's involved, it's problematic, and it's involved in autoimmune disease, periodontal disease. Chronic inflammation it's ultimately involved in the development of cardiovascular disease. Okay, and then fever. So fever is a process that is oftentimes associated with inflammation. So fever is the phenomenon by which the body's temperature increases. Fever happens as a result of the release of what we call pyrogens. An example of a really important pyrogen is IL-1. IL stands for interleukin, that's a cytokine. And what happens during fever, so we break the events of fever into these three different phases. The first one is called onset. So during onset, we get an increase in temperature. Onset happens when the hypothalamus, essentially when the hypothalamus stimulates cells to release certain chemicals. During one of the hallmarks of onset is shivering. Hold on one moment. So shivering is somewhat, or we shiver, and that it's counterintuitive that we shiver. Shivering is adaptive in the sense that it helps the body to increase temperature faster. And the reason that fever, low grade, what we call low grade fevers, are actually adaptive because they create an inhospitable environment for both pathogens and also for virally infected cells, and having a slight fever or a slight increase in body temperature helps enzymes to work better. So it helps the enzymes that are involved in catalyzing reactions that are immunological in nature. It helps those enzymes to work more effectively. After onset, we have what's called stadium. So stadium is, during the fever, stadium is when that elevated temperature is maintained. During stadium, we get, again, increases in enzymatic activity, metabolic activity is increased. Also, during stadium, the liver and the spleen bind to zinc and iron, and they kind of sequester zinc and iron, and that slows down microbial reproduction. And then the last stage of a fever is what we call defervescence. And during defervescence, this is kind of the layman's term for defervescence is having your fever break. So during defervescence, temperature returns to normal. The hypothalamus is no longer stimulated and no longer secreting hormones. And again, this is when things kind of return to normal. Okay, I think I covered most of what's on this side. So again, benefits of the fever. So having a fever, that fevers inhibit, they create an inhospitable environment for pathogens like bacteria and viruses. Having a fever, a low-grade fever, also helps to increase enzymatic activity. Importantly, it increases the expression of those cell adhesion molecules, which, as we've already discussed, allows white blood cells to migrate into the tissues via diaphoresis more effectively. And as such, it is recommended when an individual comes in, if you have a low-grade fever, it's better to let that fever play out. And that's because we as organisms clear infections faster if we can just let that fever, if we cannot treat that fever and not decrease our body temperature. However, if a fever becomes too high, that is very detrimental. And the reason it's detrimental is because at high temperatures, proteins start to fall apart and denature. And so fevers that are greater really than 105 degrees Fahrenheit, above that, proteins start to fall apart. And once we reach 109 degrees Fahrenheit, we end up, that's not compatible with life. So fevers at around 109 plus degrees Fahrenheit result in death. And above 105, again, that's where we start to see seizure activity. Okay, so that is it. No, it's not it. I wasn't able to get to this slide. Hold on one moment. Okay. So pus. Pus is a kind of the layman's term for exudate plus cellular debris. So exudate, like I said, is formed because we get this increase in interstitial fluid that takes place. And then when we talk about infection, what inevitably ends up happening is that fluid ends up getting combined with cellular debris and white blood cells. And the result of that is pus. Pus is typically removed by the lymphatic system, but if it is not, pus can accumulate and form what we call, or be a part of what we call an abscess. So an abscess is an infection that's not been cleared, and one of the hallmarks of an abscess is the accumulation, the walled-off accumulation of pus in a specific area. The walling off is actually an adaptive response by the body. Scar tissue is formed around the infection to help it or help prevent it from becoming systemic. And then with abscesses, oftentimes they are lysed by a practitioner and the pus is drained, and then antibiotics are given to help clear the infection. What's happening here? Oh, okay, so that is the left side. Okay, great. So let's move on to the second part of the immune system, during which we are going to talk about the adaptive immune response. So the adaptive immune response differs from the innate immune response in the sense that this is a response that is antigen specific. And this response is a response by our immune cells that creates memory. And we'll talk about the cells that are involved, the mechanisms by which they protect you against specific antigen, and then how memory forms as we work our way through this material. So there are two branches of the adaptive immune response. There is what we call cell-mediated immunity. So cell-mediated immunity involves T cells, And then there's what we call humoral immunity. And humoral immunity is the adaptive immune response that involves B cells and their products, which are antibodies. So how do these cells, or what are the mechanisms by which these cells operate? So again, when we talk about the humoral response, this is the response that is made possible by the activation of B cells. And activated B cells, again, produce antibodies. We call activated B cells plasma cells, and they are basically antibody-producing machines. The way in which both B cells and T cells are activated is through the recognition of specific antigen by what we call either the B cell or the T cell receptors, depending on which cell we are discussing. An important feature of B cells, and I'm getting a tiny bit ahead of myself, but B cells have the B cell receptor is essentially just an antibody that is stuck on the surface of a B cell that becomes activated. And as such, B cells are able to become activated by free antigen, right? And that is because antibodies are able to recognize free antigen. T cells, on the other hand, they also have a receptor for specific antigen. Hold on a moment. I have to readjust. Everybody still hear me? Okay, so T cells also recognize specific antigen through their specific receptors called T cell receptors. However, one of the big differences in the way B cells versus T cells recognize antigen is that T cells cannot recognize free antigen. T cells have to recognize their specific antigen through what we call, or in the context of what we call the MHC molecule. Their T cell receptor can only recognize antigen presented to that receptor in the context of MHC. So T cells do not recognize free antigen. Only B cells are able to do that. Okay, so most of this should be hopefully by way of review. So what are antigens? Antigens are, again, as we started talking about when we were in the blood chapter, think of an antigen as a cell, a cell marker, a cell identity tag. Examples of antigens are, various components, polysaccharides that are found in the cell wall of bacteria, the toxins released by bacteria are soluble antigens. The protein capsid of viruses are antigenic, are antigens. All of the cells of our bodies that are nucleated have self-antigen. Essentially, any cell that has a nucleus is going to make antigen, and typically put that antigen on the surface if it's a cell, or if we're talking about a viral antigen, that antigen is going to be soluble, right? Kind of free floating, if you will. Antigens are typically proteins or large polysaccharides, and they are what makes something antigenic is the degree of foreignness and the size. So substances that are antigenic, and being antigenic implies that it's a protein or a polysaccharide that's involved with the immune response. But being antigenic, therefore being something that's able to stimulate an immune response, requires that the substance that we're talking about, whether it be a protein or a polysaccharide, is both big enough that it can be seen by the immune system or by your immune cells, and also foreign or different enough from your own self-antigen that your immune system sees it as being just that, foreign. Which brings me to this material right here. So foreign versus self-antigens. So foreign antigens, again, antigens are used by our immune cells to tell the difference between self and non-self. And that feature is very important in our immune response. And it's very important because our immune cells need to be able to see cells that belong to us, right? And leave those cells alone and differentiate those cells from cells that are foreign, because aside from cancer cells, and actually there's a little caveat to that too, but aside from cancer cells, pathogens come from outside of us, right? They are cells, or they are viruses that infect cells that are foreign. Like I said, cancer cells are the exception to that, in that cancer cells they're our own cells that are mutated. But I just kind of want to make mention of the fact that there are many cancer cells that when they become mutated, they make foreign, I guess I'm going to use the term foreign antigen, but they make antigen that is specific to that cancer cell, and is one way that and this is one therapeutic approach that we use to try and target and eliminate cancer cells, is trying to get our immune systems to recognize if there are specific antigens associated with cancer cells, to get our immune systems to be able to see those antigens as being foreign. When we get to autoimmune diseases, or autoimmune diseases are diseases that are due to the fact that our bodies are seeing self-antigen as foreign, and as such, we start to attack our own tissues. And that is why autoimmune diseases are so difficult to treat. It's hard to get your body, if your immune cells start to see your own tissue as being foreign, it's difficult to try and train, if you will, your immune system to not see that self-antigen as being foreign anymore. And this is also the reason why inflammation is one very prominent characteristic of autoimmune disease. Chronic inflammation, if you have your body constantly attacking itself, you're going to have a chronic inflammatory process that doesn't resolve. Okay, so how do our T cells and our B cells recognize specific antigen? They recognize specific antigen by what is called the epitope. The epitope is a specific part or it is a specific site or part of the antigen that the immune system binds to and recognizes as being, I shouldn't just say as being foreign, because we use self-antigens, the epitopes of self-antigens, to tell our cells that those antigens are not foreign. So just think of an epitope as the region of that antigen that is recognized by the immune system. What makes, if an antigen is foreign or is going to be recognized by the immune system, what makes that antigen recognizable, again, is the size and the degree of foreignness. And so if something is very small, it tends to not be antigenic. If a entity like a protein or a polysaccharide has a lot of repetition to it, it tends to not be antigenic. So plastics, for example, right? Plastics are polymers. They're not at all complex. They're very simple in their molecular structure, and as such, they're not typically very antigenic. And that is part of the reason why they're so useful when it comes to incorporating them into medical devices or things that need to be implanted in the body. Our bodies tend to not see plastics as being foreign. Okay, so this term immunogenicity, so something that is immunogenic is something that is that is a word used to describe an entity that is capable of triggering an immune response. And as I keep hammering, the immunogenicity for an antigen is going to depend on its size and its complexity, or its degree of foreignness. So the larger the antigen, the more likely it is to elicit an immune response. The more foreign the antigen, the more likely it is to elicit an immune response. So then this word hapten. So haptens are really important when we start talking about hypersensitivities. So haptens are small, very, very tiny antigens that by them, very small, but very foreign. So by themselves, they are too small to elicit an immune response. However, when we are exposed to haptens, what happens is those haptens end up getting combined with our cellular material, like proteins, for example, and cellular debris. And when combined with proteins or cellular debris and then cleaned up or gobbled up by cells like macrophages and presented to our adaptive immune cells, our T cells, for example, our bodies start to elicit an immune response, because haptens, again, they are very foreign. So if they are complexed with proteins, they become large enough for our immune systems to see them. And so what happens with haptens are the types of antigens that are involved in hypersensitivities that result as kind of a secondary response. Said another and probably better way is that the first time you are exposed to a hapten, you do not have a response to that hapten. However, subsequent exposures to that hapten tend to elicit very, very over exaggerated type of inflammatory responses. So one example of a hapten is poison ivy, right? Poison ivy, the first time, and also the venom, wasp venom, or wasp and even bee venom. So the first time you are exposed to these haptens, the first time you're stung by a bee, for example, you do not have an allergic response to it, right? Or you might have a little bit of an inflammatory response, but it tends to either get worse over time, or subsequent stings can result in what we call anaphylaxis, which is this huge inflammatory response that can shut down the airways. Same thing with poison ivy. Poison ivy, the first time you are exposed to poison ivy, and poison ivy even more than bee stings, the first time you're exposed to poison ivy or poison oak, you actually don't have a response. So if you are somebody who responds to poison ivy, that means that you have been exposed, at least in some shape, form, or fashion, to that hapten in the past, right? So subsequent exposures to poison ivy or poison oak result in huge inflammatory responses. Okay, so autoimmune diseases. So here's what I want you to know about autoimmune disorders right now. So they are due to the body's, the immune cells, the T cells and the B cells specifically. These diseases come about when our T cells and our B cells start to see self-antigen as being foreign, and therefore our cells begin to mount in a response or an attack to our self-antigen, right? So I have here lacking a tolerance for self-antigen. Said another way, our T cells and B cells start to see self-antigen as foreign. Autoimmune disorders, the etiology or the origin for them is complex and is varied, right? So it can happen and does have, these diseases come about because sometimes because there are viral antigens that exist that are similar to kind of look, if you will, similar to our immune systems to self-antigen. And so when our cells become infected with these viruses, they are foreign enough that our T cells and B cells start to mount an immune response and form memory against that antigen. And then we end up with what's called cross-reactivity. So the viral antigens are similar enough to our self-antigens that when we start to make, for example, antibodies against those viral antigens, those antibodies are able to bind to and target our self-antigens. Autoimmune disease can also come about. There's very likely a genetic predisposition when it comes to autoimmune disease. So some people are susceptible to having self-antigen that undergoes essentially mutation and becomes altered enough that our bodies start to see it as being foreign and mount an attack against that particular self-antigen. You know, there's, again, varied underlying ideologies for autoimmune disease, but the overriding theme is that we see self-antigen as being foreign, and we mount, therefore, an immunological response to our own antigen. Okay, so now we're going to get in and talk about the general structure of the, it should say TCR, of the, and some of the differences in way T cells versus B cells recognize specific antigen. So as we already touched on, both T cells and B cells have receptors, right? And those receptors are specific for a particular antigen, highly specific, and that's where this antigen specificity comes into play. As I already mentioned at the beginning of this lecture, the B cell receptor is an antibody stuck to the surface of that B cell. We'll talk about the structures of antibodies later on in this lecture, but for one antibody is able to recognize two antigens. That's what you're seeing here. The structure of antibody is, it looks like a Y, right? So each one of these arms is able to recognize an antigen. So each B cell receptor is able to recognize potentially two unique or specific antigens. The B cell receptor, when we talk about classes of antibodies, is actually an IgD antibody. And the T cell receptor, drawn here, the T cell receptor can only recognize antigen in the context of MHC, and we'll talk about that here in just a moment. Additionally, in order for a T cell to recognize its antigen recognize the specific antigen it's designed to recognize in the context of MHC, it requires what we call a CD molecule. So CD stands for cluster of differentiation. T cells have different CD molecules. That's how we type them. And we are going to be talking specifically about two different types of T cells. T cells that have the CD4 molecule on their surface are what are called T helper cells. And T cells that have the CD8 molecule on their surface are what are called cytotoxic T cells. And what these CD molecules do is they stabilize the interaction between the T cell receptor and the antigen it's designed to recognize that's being presented to it in the context of MHC. Hold on one moment. Go over there, please. Okay, so that's what this slide goes over. So helper cells versus cytotoxic cells. So if I were to ask you what type of cell is CD4 positive, that would be a helper T cell, right? If I were to ask you which cells are CD8 positive, that would be a cytotoxic T cell. And one other thing I want to mention as we're on this topic is that there are other immune cells that have CD molecules on their surface. So macrophages, for example, have CD4, right? They are CD4 positive cells. And that is important because when we talk about the way in which viruses infect cells, many viruses require they require the CD molecules to get into the cell. And so viruses that are able, that use the CD4 molecule specifically to get into a cell, would be able to infect both helper T cells and also macrophages. Again, I bring that up now because we're going to talk about some examples of viruses at the end of this material. And that's an important concept that comes into play. Something else that I want to make mention of as we're talking about helper T cells versus cytotoxic T cells is that cytotoxic T cells, the word cytotoxic means toxic to a cell, right? So these cells directly destroy cells that have the foreign antigen they are designed to recognize on their surface. Helper T cells do not directly destroy antigen. Helper T cells are the orchestrators of the conductors, if you will, of the immune response. Helper T cells have very extensive cytokine profiles, And what helper T cells do is they help the other immune cells recognize and remove the specific foreign antigen that the helper T cells have recognized, right? And they do so by their very complex cytokine profiles that help and what those cytokine profiles or those cytokines do that are a part of the profile is activate the other immune cells. Okay, so this concept of antigen presentation is key when it comes to the way in which T cells recognize their specific antigen. So before we get in and we talk about the way in which antigen is presented to T cells, we need to talk about the difference between professional antigen presenting cells and just antigen presentation as a general concept. So all nucleated cells present antigen, right? All nucleated cells present antigen in the context of what we call MHC1, okay? So, and that's really important when it comes to this concept of self-antigen and the fact that our cells are able to ignore cells that have self-antigen. They're able to do that because our cells present self-antigen all nucleated cells present self-antigen in the context of MHC1. We also have 3 specific cell types that are what we call professional antigen presenting cells. Can everybody still hear me?
Yeah.
Okay, hold on a moment. Sorry you guys, this has been such a disjointed lecture, but I'm just gonna take out these AirPods because they're driving me insane. So hold on a moment. So, again, professional antigen presenting cells. These cells specifically are B cells, macrophages, and dendritic cells. And these cells present antigen in the context of a molecule called MHC2. And MHC2 is the way in which CD4 positive T helper cells recognize antigen. Okay, so I'll say that again. Professional antigen presenting cells, and the acronym for that is APC. Professional antigen presenting cells are the B cells, dendritic cells, and macrophages. And these cells specifically present antigen in the context of MHC2, and that is how T helper cells are able to recognize antigen, okay? So again, MHC1 is a molecule that's found on the surface of all nucleated cells, and it presents, and I don't wanna say it only presents self-antigen. What MHC1 presents is antigen that comes from within the cell. So all self-antigen, just by its very nature, because it's self-antigen, comes from within the cell, right? It comes from within the cell, and it's processed and presented on the surface in the context of MHC1. But in addition to self-antigen, viral antigen is presented in the context of MHC1, right? And that's because the way in which viruses work is that they get into the cell and then they insert themselves either into the DNA if it's a DNA virus, or if it's an mRNA virus, they use the ribosomes. But in either scenario, that is still antigen that comes from within the cell, right? So viral antigen, self-antigen, all of that is presented in the context of MHC1. Antigen that is gobbled up, if you will, by cells, and so it's what we call exogenous antigen, comes from outside. That is the type of antigen that is presented in the context of MHC2 on the surface of B cells, dendritic cells, and macrophages. Okay, and that's what these next couple of slides go over, right? So again, this picture kind of scares people, but really all it's showing you is that endogenous antigen so antigen that comes from within, is processed by the rough endoplasmic reticulum, and then it is presented on the surface of all nucleated cells in the context of MHC1, okay? So self-antigen, this will be the case for self-antigen. Any nucleated cell is going to present self-antigen in the context of MHC1. Viral antigen will also be presented in the context of MHC1, right? MHC1 is how cytotoxic T cells recognize their specific antigen, okay? So, and that is why when we started out, and I think in the last lecture, I was saying that cytotoxic T cells are really good, along with natural killer cells, at recognizing virally infected cells. This is the reason why. The reason why cytotoxic T cells are so good at recognizing and eliminating virally infected cells is because they recognize endogenous antigen. They recognize antigen presented in the context of MHC1, and if that antigen is viral, they will recognize that and they will tell that virally infected cell to undergo apoptosis. They will attack that antigen, right? Hold on. And I'll go back to that other side, which is the summary side, but MHC2, again, is only found on the surface of the professional antigen presenting cells. So B cells, dendritic cells, and macrophages. And these molecules present exogenous, here's that word right here, exogenous antigen. So antigen that comes from the outside, right? So we already talked about the fact that macrophages, which are innate immune cells, the way their mechanism of protecting you, or what they do, is they undergo phagocytosis, right? They gobble up substances that they recognize as being foreign. So let's say a macrophage encounters a bacteria, right? Or a virally infected cell. It will gobble up that cell. So it comes, that antigen is coming from the outside. It'll pull it in. It'll process it through the rough endoplasmic reticulum, and specifically by having that vesicle that is formed when the cell undergoes phagocytosis merge with the lysosome and forming the phagolysosome, and then it essentially breaks down that antigen and presents it right on the surface and via MHC2. That is also what dendritic cells do. They recognize, they recognize PAMPs, and then they will, pardon me, they will gobble up a cell that has a PAMP on it through their toll-like receptors, right, which we discussed, and they will process and then present that antigen. But then what about B cells? So the way B cells do this is that B cell, that IgD, that B cell receptor, when antigen binds to that B cell receptor, there is an invagination that takes place. So essentially that B cell will gobble up its receptor bound to the antigen, and it will process that antigen, and it will then present it in the context of MHC class 2. And MHC class 2, with its antigen, that is the way in which T helper cells are activated. That is how T helper cells see antigen, is in the context of MHC2. Okay, guys, and hopefully everybody can hear me. I'm gonna end right now, which I know is 15 minutes early, mostly because I'm sure this is, and I apologize for this being kind of a discombobulated lecture. I have a sick child at home, so it is COVID, super fun. But she doesn't have COVID. I mean, just COVID and having kids at home. Okay, so I'm gonna end this recording, and then we will, I'm going to end this recording, and then I'll stay on to answer questions.